Hey, it's great to be presenting to all my colleagues in Korea today. So I sure wish I could be there. And I've been to Korea many times, and I can't wait till the next time I get to come back. Let's talk about spatial biology and what it's doing to life science, cancer biology, single cell research, and many, many additional areas. It's a very exciting time right now in spatial biology. And like I'm going to give you some sort of behind the scenes information on some of how we developed these two spatial platforms today, or three spatial platforms. But when I was a professor at a university in the United States called Vanderbilt University, and I was tenured faculty there. But what I really wanted to do is to actually build instruments that could really meet some of the unmet needs that I thought was happening in life sciences, in medical sciences, and in translational research. And to do that, I really couldn't build instruments as a faculty member. And I knew I had to leave academics to actually come to a company and build boxes, build platforms that I felt had a chance to make a difference for life science research and translational research, medical health sciences. When I left academics, I started and initially I was building next generation sequencers. This was back in 2000. And I think we really made a difference. I think next gen sequencing really brought us to a new understanding of how biology works. It was the first time we ever had unlimited plex that we could measure in life sciences. And that was a big step forward. Now, the trouble with that is I had to take the tissue slice, which you see on the upper right-hand side of the screen, to get all that unlimited information and answers. You had to basically blend the tissue up, homogenize it, and then generated a next-gen sequencing result. That was really problematical because some of the rare cells disappeared when you do that. So in early 2013, actually, Nature Method of the Year was single-cell genomics, and that was a great advance. And it's a great advance because now you could see rare cells and understand what rare cells are doing. Now, the problem was with single cell was that in the end, you want to know how a tissue works and how it functions. And it functions in a spatial context. All the cells are related to each other. They're interacting with each other. They're spatially localized. And with single cell, you just lost all that information. So when I came to NanoString in 2012, I was on a mission to figure out how do we take the power of next-gen sequencing, the power of single-cell RNA-seq and single-cell proteomics, and allow us to have the unlimited plex of NGS, the rare cell capability of single-cell, but not lose the X, Y, and Z coordinates of every cell so that we could take the powerful aspects of NGS and single-cell and now spatially resolve all these materials and have an XYZ coordinate of everything that I'm measuring. So I started working hard on that, and we've been successful at that. We were able to invent and build three new spatial platforms: the Geomics Digital Spatial Profiler that we launched in 2019, the Cosmic Spatial Molecular Imager that we launched in 2022, and the Atomic Spatial Informatics Platform that seamlessly puts them both together. And I'm going to talk about all three of those today. So you could say, why did we build two spatial platforms like that? Why did we need two instruments? And the key aspects of that is that spatial biology happens on a couple of distance scales, a couple of spatial scales, and plex scales. So we really invented and built the Geomics Digital Spatial Profiler to be unlimited plex, full whole transcriptomes. For humans, that's about 19,000 fish probes that we do at once. For mice, it's 22,000. We layer on top of that about 150 proteins that you can do simultaneous. And geomics was designed to actually measure unique tissue histological features that, in my mind, generate function. There is no single cell organ in your body. Multicellular, multiple cells come together over a spatial domain of around 50 microns out to a couple of millimeters. And those are the things that actually generate physiological function in your tissue organs. And that's what we designed geomics to do. Whole transcriptomes of these histological substructures that make function. Now, these histological subfeatures are made from individual cells, single cells that come together and organize. And sometimes you need to actually go in at the single cell level and even subcellularly and understand exactly all the conversations, all the communications that's happening between the individual cells. How are they sharing information? How are they talking to each other? How do they communicate? What cell types are they? And that's why we invented the cosmic spatial molecular imager. So 2 platforms, one functional at the sort of whole tissue, multicellular, histological subfeature functioning aspect, and then the cosmic spatial molecular imager at the single cell level. And they're both needed to have a complete understanding of how tissues and how organs work. This sort of summarizes that. Let's start at the bottom. The geomics, multicellular, whole transcriptome, 20,000 plex, 150 plex of protein on the same slide, and a high throughput. So you can actually look at entire cohorts of patient, relatively simple, 120 samples a week. Now, the cosmic spatial molecular imager, again, it's answering the private lives of how individual single cells talk to each other. We're at 6,000 plex in RNA imaging now, the largest in the world this time, 120 plex simultaneous protein, but at lower throughput at the level of sample number, but ultra high throughput at the level of single cells. You're getting about 5 million single cells every time you push go on a cosmics. So I think it's important when you're thinking about using or utilizing a couple of platforms to understand, well, how are they born? How are they generated? And this is a scribbled note. This was literally, I was waiting to give a talk in January of 2015 at the MD Anderson Cancer Center. And what you see there in the center is literally the notes I was taking and scribbled out a chemistry that would allow me to have unlimited plex on biobanked FFP tissue samples and still have spatial context. That was the heart of the beginning of geomics, was January of 2015. It was just a note on a pad of paper while I was waiting, scribbling out in a hotel lobby as I was waiting to get picked up. And later that day, one of the key investigators I worked with at MD Anderson, he said to me, Beechem, don't invent a fancy high-plex microscope and imaging device if it can't work on real-world archived biobank samples, medical grade, clinical samples that come out of a hospital, because that is where the medical history of the planet is. And we need these tools to work on those types of samples. So I really took that to heart, what Gordon told me that day, and everything that we do on both the geomics and the cosmic spatial molecular imager, every design decision that I made was a design decision to allow those instruments to generate all this high plex information, but to be able to accomplish it on real world archival biobank samples. So that's, in some respect, that's how geomics and cosmics were born, with that sample type in mind. That's really unusual because almost none of the other techniques were born that way. Okay, now the cosmic spatial molecular imager was happening at the same time we were inventing a new set of optical barcodes. But in this particular case, we actually, I was interacting with a semiconductor company, tools-based company called Lam Research. They were located down in Silicon Valley and in California here. And they were thinking about how to get into life science research. And they funded my laboratory here at NanoString R&D to $50 million USD back in, you can see that August 8th of 2017. And that allowed us, that gave us the freedom to build the world's smallest information content imaging optical barcode. But again, the same idea, we wanted to image in clinical grade samples. And that is the heart of how cosmics was formed. It actually started out as a hybridization-based sequencing technology called hide-and-seek, but its final incarnation of that chemistry is actually to go in situ in biobank samples and allow us to do unlimited multiplexed imaging of those samples. So that's how those two platforms were born. And I think it's important to sort of remember where they're coming from. So let's talk about the chemistry we use. And again, we design these chemistries with one thing in mind. They work on archive samples, but they also follow standard pathology workflows. I'm not clearing tissue and vetting tissues, doing all these other things that a lot of the other high-plex imaging technologies do. They work on a normal 5-micron FFP slide. All the reagents are in situ reagents. That's one of the key aspects of if you're doing spatial biology on archival tissue, is you can't take the material out. You got to leave the material in. That's why all our chemistries are in situ. And over on the left side, you see our fish probes are just normal 35 to 50 base pair hybridizations with the RNA that's intrinsically in the sample. This was the inventive thing that I scribbled down on a piece of paper and MD Anderson. Instead of putting fluorescent dyes on these fish probes, which everyone else was doing at that time, I just put a little photocleavable linker and then a sequencing index. And what do microscopes do for a living? They shine light in tiny places, and I just used the microscope as a light source. I put 20,000 fish probes at a time, and then the light actually localizes which fish probes I'm looking. They photocleave the little photocleavable sequencing barcode, and I read it off on a sequencer. I got a high resolution image, and I just put those two things back together. We do the same thing on the back of antibodies. I site specifically put an oligonucleotide on the FC region of an antibody with a photocleavable linker, and that's how we get unlimited plex on proteins and fish probes. Now for cosmics, we need to subcellularly localize those particular signals. And to do that, you have to build an imaging barcode. So I take exactly the same chemistry that we're using over here and for the cosmics, or for the geomics, I'm sorry. The business end's the same, 35 to 50 base pairs of hybridizable unit, except now instead of a photocleavable linker, I have a sequence, I call it a 4 landing pad piece of DNA, and it is home to hybridization reagents that are the special optical barcodes that we made that contain all this information. They will actually hybridize to the tail of these oligonucleotides. You cyclically image them. And with 4 colors and an optical barcode, I'll show you, we can design to go up all the way to imaging in a whole transcriptome. Right now, we are at 6,000 plex, the largest in the world imaging of in situ RNA. Similarly, for the proteins, similar type of cyclic measurements that we make. And we are also now the world's leader on imaging antibodies and at subcellular localization. We're at 120. Now, it's important to remember the chemistry that the design of the probes that we do here actually are common, not only with our spatial, but they're also common to our non-spatial nCounter technology. Our nCounter technology has now been taken all the way to the FDA. There's bona fide clinical assays running on the nCounter assay. And it's important to remember the hybridization probe design and everything is identical between FDA cleared, even RNA-based assays that we do now, all the way, it's common to both cosmics and geomics. So you can think of these technologies all in a similar kind of manner, in a similar kind of properties. Okay, you don't have to take my word for this in terms of how's it work and does it work and all those. Both of our spatial biology technologies been accepted on the highest standards of peer review. As Nature Biotechnology, May 2020, published our geomics digital spatial profiling. And then on the right, you see in December, actually it was first on, but October of 2022, was the Cosmic Spatial Molecular Imager also accepted and peer reviewed at Nature Biotechnologies. This gives you an example image of the geomics looking at a single human lymph node, and then 5-micron section right under that run with a cosmics, and that's full-blown imaging of a whole lymph node, 1.4 million cells here at a few thousand plex, and then here the geomics is looking at these germinal centers at the full 20,000 plex. but these information contents really work together well. Now, here's some fun facts that just happened recently this year. We ended up making that cover issue. The Cosmics actually became the cover of the December print issue of Nature Biotech. And the only thing they say at the bottom is spatial molecular imaging. That's our stuff. And that's our own words that coined that and made the cover. Now, when I was doing this work, and that work was being published in Nature Biotech, believe it or not, the National Geographic had heard that we were actually now imaging a human brain at 6,000 plex. They actually contacted us. They were running an issue you see right there in the middle on dementia, Alzheimer's disease, a whole issue on that. And they wanted to have an image of 6,000 plex human brain in the National Geographic. So we were able to do that. We were able to submit this image. It made it into the January 2023 issue of National Geographic. So kind of first time I've ever had sort of an image like that in a popular science magazine. So, but it was because it was so important, so new. We were addressing an important problem, which is neurodegenerative disease in the brain, that if we all live long lives, we're probably going to end up with this. So research there is very important. So let's talk about the Geomics Digital Spatial Profiler here. The whole transcriptome, highly sensitive, cost-effective, any type of sample. FFP, it was all designed for FFP, but it works with fresh frozen whole mount, tissue microarrays, organoids, fixed cells that can pretty much measure almost any sample you can put on a microscope slide. It's multi-omic from the beginning, RNA and protein all in the same section, and high throughput, 120 samples a week. I described this chemistry, so I won't describe it again, but this highlights the important part of light and how we get whole transcriptomes that are uniquely shaped to the sample that we're investigating. The key is in the back of the Geomics Digital Spatial Profiler are two semiconductor chips about the size of your thumbnail. And when the Geomics takes a picture, a camera image of your tissue, and every tissue is different. I've never seen the same geometry twice on any of these things. The camera takes a picture, then these little semiconductor mirrors adjust to the exact geometry of that particular tissue slice. And then when we want to get the whole transcriptome out of that, those little semiconductors are just mirrors. You can see the light comes in underneath the sample, and they are shaped to exactly the tissue shape with single cell resolution. The light comes up, photocleaves the whole transcriptome in a particular area, and that's where your signals come from. So I call this is the geometry of cancer. And this is one of the reasons why you don't want to blend your samples up in a blender and then just sequence them, because the unique geometries, and you're looking at non-small cell lung cancer, the green cells are the tumor-specific cells in this particular sample. The immune cells are lighted up yellow. Specific immune cells, in this case, macrophages, are lit up light blue. And then the rest of the non-immune content in purple. So when I put that particular sample in, there'll never be another non-small cell lung cancer that looks exactly like that. That's linked to that patient. The camera will take a picture. The mirrors will immediately flip, and they will match the geometry, say, of the tumor-specific cells. I reflect the light in, comes in, it hits only the tumor. The whole transcriptome of the tumor-specific cells come out. That whole operation takes about 30 to 45 seconds. Those photocleave barcodes go into one element of a on-instrument microtiter plate. And then the mirrors change. And then we look at, say, CD68, the macrophages. Light comes only on those particular single cells in there, and I get a whole transcriptome with a macrophage. And then I go and do that for the rest of the immune cells and the stromal cells. Each one of those operations takes about 45 seconds. And then I just collected whole transcriptomes on those three unique pure spatial components. And then I wander through the tissue generating all this high plex information. It never was done before, and we're still the only chemistry that can do that type of operation. I think it's interesting to actually go back. I went back and looked at some of our first experiments of this type. This was actually one of our early ones. We're working again with MD Anderson. This is before we're commercial. This is on the right side is the very first biomarker response graph I ever made on a clinical trial, and they're running it. On the right are biomarkers. These were protein biomarkers then that were enhanced in responders to checkpoint inhibition in melanoma. And on the left-hand side are enhanced in non-responders. I circled a couple of unique ones because when we discovered this, MD Anderson thought we made a mistake. They said, you know what, I know the B cells aren't really contributing to this checkpoint blockade, and the checkpoint blockade's really targeting T cells and things that. And I said, well, we should look at the images and make sure that maybe the data is telling us something that we didn't know about. It turned out it was telling us something that important that we didn't know about. It told us, and we discovered with this, a thing called tertiary lymphoid structures in tumors. It turned out to be the most predictive histological subfeature in tumors that can predict response to immunotherapy, and we discovered it this way. We were able to power up to a couple of hundred patients, and we ended up with back-to-back papers in Nature from two independent groups. One was MD Anderson, the other one was a group from Lund University. All these studies were run in my lab before the genomics was actually commercialized, but we ended up with back-to-back papers in Nature, making this fundamental discovery. And the reason we made that discovery was that we were the highest plex working on archived clinical trial samples, and we made that discovery in this manner. Now we have over 250 papers in genomics right now. We made the covers many, many times. And we continue to advance the technology forward. When we introduced whole transcriptional profiling to the genomics, then it took off to a whole new level. High sensitivity, similar kind of, you can use the mirrors in just the same way with the whole transcriptome, up to 20,000. And the key thing here is that you can do these at scale. So the beauty of the omics is that I can take an entire cohort of patients, say 120 samples a week. You run them through an automated Lycobond process if you have a tissue processor, all hands off. You get whole transcriptome information spatially resolved. There's a push button, whole transcriptomes interact with Illumina for their next-gen sequencers, and you end up with whole transcriptomes and high-resolution images. You do 10 times more whole transcriptome throughput than any other platform on the market now on these key archive samples. So this is a whole new way of doing translational research. So we actually interacted. One of the papers we published not too long after we did this was a consortium of investigators working at 13 medical centers around the world, 17 investigators. And they all agreed perform this type of doing translational research on these archive samples in breast cancer. They laid out exactly a roadmap of how you should accomplish these types of studies. First time that's ever been done with this kind of high plex technology for translational research. Now we've continued, now we've expanded what we're doing in the genomics in terms of the spatial atlasing work. We did our own little spatial organ atlas, and if you want to just wander around, the NanoString people that are there will be able to tell you how to look at on a pathology guided tour of whole transcriptomes in tissue. I recommend that. Now we scaled this up. We actually announced at AGBT this last year a comprehensive spatial atlas of 30 healthy human tissue organs of a variety of demographics to provide a baseline study for all your spatial work. And then we had a real big announcement that we literally made just June 2nd, 2023, just 2 weeks ago, a week and a half ago. We paired up the genomics technology, paired up with an artificial intelligence machine learning company called Alkin, and paired up with a whole bunch of medical centers from around the world. We are going to be looking at 7,000 tumor samples at this high plex using both genomics and cosmics. We'll be applying artificial intelligence and machine learning to actually understand some of this high dimensional information and really try to transform how health science understands cancer and then also treatment and diagnostic. 7,000 patient study on geomics and cosmics married up with AI and ML. So I think this has the potential to be completely transformative to our understanding of cancer. Okay, I'm going to end on just a little note here. I can see I'm running just a little bit slow. But our high-plex assays in geomics at the whole transcriptome are as repeatable as our clinical grade FDA cleared assay for Prosigna, which is a breast cancer 50-plex. This is not a high noise technology. It's absolutely very, very reproducible. We've actually had some people start taking the geomics into the CLIA lab, the night diagnostics lab, who you see on the left. Is probably further along than anyone else. They validated over 100 of our barcoded antibodies and actually used those directly in their clinical trial work. And then on the right, I'm just emphasizing that we've been all the way to full-blown clinical assay work and FDA cleared diagnostic from our nCounter technology. And just remember that the design principles of nCounter are identical to our design principles in both cosmics and geomics. Now, it works not just on cancer. This is what the geometry of diabetes looks like. I can't talk about that, but you can actually see early diabetic phenomena, things related to metabolically healthy obese patients, and so on and so forth. This work is all, I think in bioRxiv, you can find some of this work. And also developmental biology. You can actually put mouse embryos into these geomics and then generate an entire whole transcriptome of organogenesis and developmental biology. So all these fields can be implemented in this high-plex manner. So now let's talk about cosmic spatial molecular imager. Okay, highest plex that anyone's doing in the entire world right now. We're at 6,000 plex, over 120 plex in proteins. Again, it works on any sample. Archived FFP, it was designed for that, but it does fresh frozen TMAs, organoids, fixed cells. It's multi-omic on the same slide. Tunable output from 2 to 20 slides a week, but it's really 5 million single cells every time you push go and run. And it's high resolution, subcellular single cell resolution, 50 nanometers, 50 nanometers in X and Y, that you're localizing these individual events. There's the cosmics, what it looks like. There's the chemistry that I already talked about. Let's see what some of these things look like. Every time you push go on a cosmics, you're measuring roughly 2 to 4 million single cells and about 3 to 6 billion transcripts per run. You can think about it, every time you push go, you're measuring basically on the order of 10% of the entire human cell atlas in one run of a cosmics. You could also think of it, this is like in situ sequencing at a NovaSeq 6000 level. That's the 3 to 6 billion transcripts, but now they're all spatially resolved. Spatially resolved means you never have to dissociate the tissue. This is what tissue looks like in a cosmic spatial molecular imager. And people studying that now at high plex, they actually do all these things to try to dissociate these cells out off of these samples, out of these samples, single cell RNA-seq. And you know what? You cannot dissociate these kinds of tissues with a high fidelity and high efficiency. When we measure them in cosmics, there's no cell left behind. Greater than 98, 99% of every cell that you put into the cosmics, you're going to get full-blown high-plex RNA and protein data from. So if you think about dissociated single cell work, typically you'll have 10,000 cells per sample, 50 millions or so transcripts. Exactly that same sample in a cosmic is going to give you about 1 million cells and multiple billions of transcripts. No tissue loss, intact tissue section. So I really recommend, if you actually are dissociating some of your hard-fought FFP tissues, biobank samples, just measure them in situ. You get 100 times more cells and you'll get nearly 100 times more transcripts. Okay, so what's it look like? That's the cosmics. When you're doing single cell, your end of your analysis is that thing over on the left. Those are the UMAPs of the single cells. And cosmics is just the beginning. So you actually go from, that's a single human lymph node that you're looking at. And you go from that UMAP, they all got X, Y, Z coordinates, and you see that beautiful lymph node structure on the right-hand side. There's no matter what bioinformatics I do on that UMAP, I would never predict the beautiful lymph node you see on your right side. So you got single cell and subcellular resolution. Every single dot you see is a single RNA located in space at subcellular resolution. It's really hard to comprehend the magnitude of the data that comes out. So instead of showing you 4 million cells worth of data, I'm going to show you basically 6 cells of that tissue that we're doing 4 million of. And what you see is in 3 dimensions is 6,000 different colors of mRNA. Those big round glowing sort of gray balls are a single nucleus stained with DAPI. But you can see how we're measuring now all the RNAs wrapped around that nucleus and inside the nucleus. And you're doing it on millions of cells at a time, billions of transcripts. Now, when I was making this, everybody else in the field was saying, okay, I think we're going to do low plex spatial biology and try to use all the single cell RNA-seq and marry it up with my low plex spatial work and try to understand what's happening. I wasn't satisfied with that at all. I wanted to get all the spatial biology information out of one 5-micron bioarchive sample. So this is a Bernoulli graph of sort of the reactome. How many things can you see in the reactome? The green is what you're measuring if you're at low plex, like 250 plex, which a lot of the other groups are at. And the white means I don't have any signal from that. We launched with 1,000 plex. We felt that 1,000 plex was what you needed to generate a general enough panel so I can measure many, many samples. And that's our current 1,000 plex that we're doing in spatial biology. No one has a commercial panel of this size. But I still wasn't content with being able to measure at this 1,000 plex. So we've now advanced the technology to 6,000 plex. So with a single 5-micron slide, biobanked archive sample, I don't need any single cell RNA-seq. I'm measuring all the biology. You can see I almost cover up completely the entire reactome on what I'm measuring spatially now, and you get it out of one slide. It's really hard to conceive of 6,000 genes, so I just took a snapshot of the Excel sheet that houses these things, and you can just look like take a look at the inside. There is something like, okay, I'm just interested in, say, cell cycle events important in cancer. I got 486 genes linked just to cell cycling in there. I got 324 genes for epigenetic modifications, so on and so forth. It's a tremendous amount of content that you're looking at the same time. Now, a lot of people say, could it work? So I've done this already in geomics. So I know how whenever I make a high plex panel, I embed the lower plex inside of it. So then if I go to high plex, and you can see on the left, I'm at 6,000. I'm imaging 6,000 RNAs subcellularly. And I embedded the 1,000 commercial plex inside that. And if the 6,000 works and doesn't bother any of the 1,000, I just end up with a straight line for the 1,000 that are common between those two experiments. And that's what you observe. So we're doing 6,000 plex. It's just as robust as our 1,000 plex, except now you have all this extra content, and you also have extra transcripts. You can see your counts per cell. We're really the only imaging technology that's now broken what I call the 4-digit. We are over 1,000 transcripts average per cell on biobanked archival tissue sample. You can see liver cancer there at 1,382 average transcripts per every cell that we're measuring. And then these are some of our control reagents that we use to debug the system, over 2,000 transcripts per every cell there. So does it work on real samples? So that's an important thing. So I can do all this in the lab, but does it work? Some of the first samples we looked at, we just made the cover of Nature Genetics studying pancreatic cancer. So I paired up with this investigator. Actually, we're going to hear this talk, I believe, later in this session. Will Wong, an incredibly talented investigator, will talk later in this session. I'll just highlight a little bit of what we discovered there. We're the first ones to ever do 6,000 plex in cancer like this. And this is what we found. Again, I'm going to let Will tell you all about it. The key thing for me technically is when we went in and measured that, by the time you see about 50,000 cells of these pancreatic cancer, one of the most difficult ones to measure, I had over 5,000 genes out of my 6,000 plex panel yielding signals 3 standard deviations above our backgrounds. So all the biology's in there. You just got to go in and measure it, and it's there. We did 8 plex on there. You see out on the left side, protein, simultaneous with 6,000 plex RNA, and you can see the densely packed transcripts we got in these pancreatic samples. And then I'm going to go quickly here because you're going to hear it from Will. You can do some analyses that are unheard of. We did spatial correlation of all the signaling ligands in this particular sample. It's 407 by 407 ligands. That's 160,000 ligand correlations we can look at in space and see how far our cytokines and chemokines signaling in intact pancreatic samples. I think we're one of the first groups to ever measure that. You see it on the right-hand side, from 30 out to about 120 microns of localized in situ signaling happening between a variety of cell types and signaling ligands. The one I highlight in the middle was one we didn't expect, macrophages and malignant cells. But look at over there, nerve cells are actually an active signaling component of these pancreatic cancers. And we discovered that because we had all this biology in our samples. Now, Will's going to tell you more about pancreas. Some people say, well, pancreas is weird. So we measured it in liver, got the same kind of results, 5,000 signaling molecules that we're measuring. We were able to correlate and determine the in situ ligands that are signaling in liver and liver cancer. And you can see a whole bunch of HLA ligands here, a variety of conversations between different cell types. And we went into the brain. Yeah, turns out signaling like this happens in the brain. We were able to see 434 squared different correlations back and forth. You can see the pathway maps of all the different types of conversations that are happening here. We had one that was really interesting. Yeah, over on the left, that is a particular cell type in the brain that's only talking to itself through these ligands called SSD, CORT, and NPY. And no one knew what that was. We had the image of that particular cell, and when we showed it to our neuropathology people, they said, oh, Beechem, that's an SST shuttle neuron in the brain. It's one of the rarest of the neurons, the largest projections. And you can see, I didn't have to make a custom assay for that. When you're at this kind of plex, the tissue itself makes its own assays. It highlighted this unique property of that cell. I didn't have to know it beforehand. The biology and the spatial localization of this high plex told me it was there. And you can see all sorts of new cell types that way. Now I'm going to end on what we do with proteins. As we advance the RNA, we are advancing the proteins. We advanced, and our protein panels on Cosmics are all debugged, validated panels that work together. You never have to worry about that. You get incredibly beautiful high-plex protein images. This is actually a piece of a archived colorectal cancer here with a little Peyer's patch right there in the middle. And I'll show you one example of a clinical result here because I think it's very unique. We were working with Oregon Health Sciences. That's a single core needle biopsy of a breast cancer sample, a breast cancer patient there, and I'm blowing up a little region. This is a breast cancer patient as part of an experimental immunotherapy plus tyrosine kinase inhibition study. Now, we can measure the phosphorylation status of a whole bunch of proteins. I'm showing you phospho-ERK 1 and 2. When that molecule is activated, it travels to the nucleus, where the only imaging technology can watch that translocation of activated ERK go from the cytoplasm to the nucleus. And we measured, yes, that patient had activated nucleus, nuclear phospho-ERK doing cancer signaling there. That patient was given a tyrosine kinase inhibitor. A couple of weeks later, we went in and looked at another corneal biopsy from that patient, and all the activated ERK is gone. So it's a direct visualization of that particular therapeutic intervention in an experimental clinical trial we were able to observe. Something only protein can do. And we're now doing it 120 plex. These tissues are beautiful under all these types of imaging conditions. We got a special emphasis on the brain, where we're doing all sorts of interest in imaging in the brain, neurodegenerative disease, and a variety of others. And then we put all this information together in a spatial informatics platform called Atomics. So cosmic data, these are big data sets. The instrument can stream the information up to a cloud that allows you to instantly share and collaborate with all the investigators around the world. You never have to move your data sets anywhere. High security, cost-effective way of handling this high spatial data.